first finish up some aspects that we left undiscussed last week concerning the soil fertility evaluation. And then after, whenever that is over half an hour or so, we'll proceed with our midterm exam. So we discussed last week really a whole variety of different strategies to look at soil fertility. What are very rough and general strategies to look at, analytically look at soil fertility? Can we just maybe have a brief shout out, a brief strategies? Helen, what's one idea? Sorry? Cat and exchange capacity is an important one. What are other strategies? So you're looking at the soil here. Other strategies?
Measuring mineralization or what have you, different nutrients.
So maybe a release of an unavailable form into an available form. So flux, a transformation, is another important category of looking at soil fertility. Yeah, what are others?
Like the plant itself, looking at how it's growing.
Yeah, so not looking at the soil, but looking at the plant. What are their two different ways that you can approach that?
We can do you can just look at the plant itself, like visual stuff, or you can take plant tissue tests and run them.
Yeah, so chemical analysis of contents versus just a visual analysis. What does the plant look like? Yeah, so I think that this is a good start. And very often we have problems, however, to really nail down deficiencies or toxicities in the field in a given location. Let's say we're going out into the field here on a farmer says, my crop doesn't grow. Or somewhere in Cameroon, you go into the field and it doesn't grow. Maybe you can discover there is it looks yellow, it looks red, it's growing very weak. Maybe you can even make a soil test and find out, well, it's sort of low in phosphorus. But very often it's very hard to find out what is the really critical nutrient, what's the real critical process that limits plant growth. And then controlled experiments, bioassays or field experiments are necessary to really nail down what the issue is. And what I called here bioassay is just that you make controlled experiments with treatments and look at plant nutrition, crop growth, but you make an intervention. You fertilize or you add manure, and you can do that very controlled in the laboratory. You can do that here semi-controlled, where you allow ambient moisture and precipitation and temperature, but still relatively controlled because you can mix the soil and control more or less for soil variability, or you can do it in the field experiments as well. So this is an example from the Ivory Coast, where different soils from an intensive fallow experiment were used. And here you see these are all different soils that came from sites under different intensive fallow, woody intensive fallow. You might have heard about Glycidia sapium. You might probably heard about Leucaena leucocephala. Albizia guacepele is an important plant tree in Western Africa. And then you can crop, for instance, an indicator crop here. They just used rice to look at the growth and the mineral nutrition. And then we are also able to look at transformations. Here, for instance, the N mineralization was found to be linearly and fairly well correlated to the rice dry matter. So nitrogen was an important limiting factor that could be improved with these improved fallows. And here, for instance, this Leucaena collinsi appeared to have been very efficient in improving nitrogen availability by increasing nitrogen mineralization in the soil. So that's an important way of getting at soil fertility. Here are some other ways of doing that in pots or in fields. Here, what they call a simple plot or container trial. Can someone of you interpret that? So these bars, it's just hypothetical yields of a field A, a field B, and a field C. So what would you think in field A? The minus n, and plus n is increased. So what can you walk us through, Tara? So in field A, nitrogen is probably limiting. In field B, is not limiting. And what's happening in field C? It's something else. It's not nitrogen that's primarily limiting. Yeah. So you wouldn't have found that out if you just go in the field and sample the leaves or the soil. It would have been very hard to tease that apart. Here with what they call a subtractive trial. Can you walk us through that? Pete, Mary, can you walk us through that?
Well, it looks like they take the overall yield and then they sort of piece out the different nutrients to see which one could be limiting.
Yeah. So which one do you think is limiting crop growth the most? No. Nitrogen, yeah. So when you take away nitrogen, when you fertilize fully, you give N, P, and K where it says all, and it grows maximum. And then you only give P and K on no N, the yield drops the most. Whereas if you leave out K, it doesn't matter at all. So also that, it would be probably very, very hard to find out these interactive effects of nutrients, very hard to find out. Here is even more complicated trial, what they call a factorial trial. Angela, do you want to walk us through that? So obviously this is probably what they think is the control. That didn't give anything here. They gave only nitrogen, only phosphorus, only potassium. Here NP, NK, PK, and finally NPK. So what do you think is, which nutrients are limiting and which nutrients are not limiting in this soil for this crop?
Well, first I would look at NP and NPK, and they seem to be pretty equal. So I would say that potassium is not limiting. Then you look at like NK and PK, and they're about the same. So I would say that probably nitrogen and phosphorus are having almost the same effect on yield.
Yeah, so nitrogen and phosphorus are probably both strongly limiting. And what about potassium?
I would say it's probably not very limiting.
Yeah, I think you're right, because whether you add to the NP, you add K, doesn't make any difference. But N alone didn't really cut it. P alone didn't cut it. Only N and P together really made the difference. To fertilize potassium with nitrogen didn't make any difference to have the nitrogen alone or the phosphorus alone. So also this would have been very, very difficult to find out that you need N and P in this situation and not K without making really a factorial trial. And you probably, most likely, would not have found that out just by doing soil analysis or just by doing foliar analysis. You really need these bioassays, and they can be very small. They can be just a little pot. They can be a big pot outside. They can be a whole field. But I think it makes a lot of sense to try it out this way. It's also a very cheap way, actually. It's much cheaper. For instance, if you look at tropical countries where maybe infrastructure and ways of doing chemical analysis are reduced, to do a small pot trial somewhere in the middle of nowhere with just any grain crop that comes in handy there is very easy, and fertilize what they have and make a good standardized pot trial is very easy rather than trying to do chemical analysis. That way you find and just record yield, just record biomass growth, maybe even only height or counting the grains. So very easy way of assessing soil fertility and a very effective way in many circumstances superior to just foliar analysis and and nutrient analysis in the soil without a trial. However, you will probably not find out much about the processes controlling availability. So that's where the limitations are. It's a good way of screening which nutrients are limiting, but understanding more about the processes, why they are limiting, and more subtle interactions, for instance, between organic and inorganic fertilizers, is not easy to tease out of these data. Additional strategies and ways of thinking about additions is we of course not only add a certain amount, but we have also the option to add a little bit more, or a lot. And very often there's a response curve. We had talked about that already, that if you add a very little and increase that, you probably increase your yields, and then it plateaus at some point, and then you might even encounter toxicity if you add a lot of a nutrient. And we have even seen for a nutrient like nitrogen, there's even toxicity possible. And there's probably somewhere a sufficient amount where you say, doesn't really matter if I apply more than that. But that's not the whole story. Here, for instance, if you look at sugarcane yield, you'll see that, yes, the sugarcane, the cane yield plateaus probably here. So you would say you would like to apply nitrogen to an amount that the sugarcane yield is highest. But actually the sugar content and the sugar yield declines at that point already. So you also need to not only look at maybe total yields, but maybe also at yield quality or the precise product that you are interested in. Same for grass yield, and going exactly the opposite way, the forage weight plateaus probably at a lower addition of nitrogen than the protein content. If you're interested in high quality grass and high quality fodder, you might need to fertilize more than the actual yields in tons per hectare would recommend to you. Then we have situations here. Where with the interaction between different growth limiting factors, you add nutrients from low to high, and nothing happens because something else is limiting, or this response curve appears that we have talked about earlier, or that nutrient is not limiting anymore, but also nothing else is limiting. You have already reached your highest potential of yield. Again, a different story is about costs. It's not necessary that with the highest yields you get also the highest returns. You see here with the nitrogen applied, and nitrogen is costly, and all fertilizers are costly. Maybe less so in this country than in other countries. But you probably have a more or less linear increase in costs. The higher the application is, that might not always be true. But let's, for simplicity reasons, assume that it increases linearly, and you get a overproportional increase at low increases of nitrogen applied, and then it plateaus somewhere around here. You would probably say for maximum yield, you would like to apply this quantity of nitrogen. But actually, the margin between the returns and the costs are higher at a lower application of nitrogen. And if you even figure in some environmental factors that are not figured into this costing, then you might even go for a much lower application rate. So only looking at biomass and yields might be misleading in this respect. And it unfortunately gets also more complicated. We have already seen a little bit the interactions between different nutrients, but they're not only nutrients that limit growth. What are other factors that limit growth? I mean, in this class we concentrate on nutrients, but what are other factors? Just shout out a few. Debbie, what are other things that could limit yields? Competition between plants, yeah. Other things, other minimum factors that are there.
That's
Pests, water, temperature. What are other things?
improper variety grown. Improper variety.
So the wrong variety for this condition. Yeah. So sorry. Say it again. Poor stand. Yeah. That's another one. Yeah. So we can come up probably with a whole list of other issues. And sometimes there's an interactive effect between additions of nutrients and these other factors. Here, for instance, with the variety, if we increase our nitrogen additions, the grain yield in the improved variety might nicely increase over the whole range of our nitrogen added. But the old variety might actually not benefit at high application rates. So whether you have an improved variety or a suitable variety, it sounds always that this is the new and the improved is the better. But sometimes actually the old ones are quite good and can very often exceed yields at low application rates for the improved varieties. Here another one, the interaction between water. We can probably increase our nitrogen contents a lot and get very poor returns for it if we neglect that the crop might actually need water and is constrained by water. So we need to look at that as well and have that in mind. Weed control is another important factor. Weeds actually also like nitrogen and phosphorus, and they are very, very efficient in competing for nutrients with the crop. So very often what we find in very fertile soils that we have also a lot of weed problems. So we need to control those weeds, especially when we add nutrients. The difference between weed controlled and no weed control is very low at low nitrogen and phosphorus additions. But as you increase, it could dramatically increase. So we need to have an additional management strategy, in this case for weeds, to make really use of our nitrogen and phosphorus additions. And then we have said that already there can be interactions between different nutrients. And here shown is the interaction between nitrogen and phosphorus. Apparently here, if you increase the nitrogen in this scenario, the yield goes up only a little bit if you don't apply P, and stays the same or even slightly decreases for highest additions of phosphorus. And only if you add phosphorus at the same time, you get a satisfactory response for your nitrogen additions. And there might be a threshold also. It makes already a huge difference in this scenario to apply 50 kilograms. To apply 100 instead of 50, that means doubling your expenses also, makes only maybe another 10% increase. So also there you might be interested to know, do I need to really apply 100? Yes, I get more out of it, but is it really worth it, this little return here or not? So these are very important factors to consider, and those you probably will only figure out in field trials. And this is the last thing I want to quickly highlight, and this is actually just a placeholder for what we discussed a week ago when Julie was here, that in a lot of the traditional or farmer perception of soil fertility, and this figure or this table highlights this from a work in Rwanda, I believe, that soil fertility in their assessment was the most important criterion for classifying soils, and they did that by looking at crop development and yields and content of soil organic matter. But there were a lot of other factors that were perceived or assessed by the farmers, but were found to be much less important for their classification of soils. So it seems, of course, what the farmer wants out is always crop growth, and it seems the chemical fertility for them was the most important, even before something like water availability that they could maybe assess by depth or texture. This was it for our part on the soil fertility evaluation. And now we switch gears and we'll look at the midterm exam.